How neuromuscular transmission occurs. Neuromuscular transmission, explained simply. In today's video, we're diving into one of the most important physiological processes in the human body, neuromuscular transmission. This is the mechanism that allows a nerve signal to turn into a muscle contraction. Without it, we wouldn't be able to walk, breathe, talk, or move at all. Section 1. What is a neuromuscular junction? U30 or 130. To understand neuromuscular transmission, we first need to know where it happens. The process takes place at a special connection called the neuromuscular junction, or NR. Think of the NMJ as a meeting point between the motor neuron ending, also called the nerve terminal, the muscle fiber membrane, called the motor end plate. These two structures never actually touch. There's a tiny gap between them called the synaptic cleft. Inside the nerve terminal, we find vesicles filled with acetylcholine, EC, voltage-gated calcium channels. On the muscle side, the motor end plate has folds in the membrane to increase surface area. Now that we know the setting, let's see what actually happens when a nerve fires. Section 2. Arrival of the nerve impulse, 130-210. Everything begins with an action potential traveling down the motor neuron. This electrical impulse moves along the axon like a wave until it reaches the nerve terminal. When the decision point arrives, the electrical charge inside the nerve terminal changes. This depolarization opens voltage-gated calcium channels. Now calcium, which is normally higher outside the cell, rushes into the nerve terminal. This calcium entry is the key that unlocks the next step. Once calcium enters the nerve terminal, it triggers vesicles containing acetylcholine to move toward the membrane. These vesicles fuse with the nerve terminal membrane through special proteins called snare proteins. As the vesicles fuse, acetylcholine is released into the synaptic cleft by a process called exocytosis. Millions of ACH molecules are released in a fraction of a second, quickly diffusing across the tiny gap and heading straight toward the muscle. Section 4. Binding to H receptors, 305. On the muscle side, the motor end plate contains many nicotinic acetylcholine receptors. When H binds to these receptors, the receptor changes shape and opens an ion channel. This allows sodium ions to rush into the muscle cell and a small amount of potassium to move out. Because sodium entry is much greater, the muscle membrane becomes depolarized, creating what we call an end plate potential. If this end plate potential is strong enough, it triggers voltage-gated sodium channels on the muscle membrane to open. This generates a muscle action potential, which spreads along the entire muscle fiber. From here, the action potential causes calcium release inside the muscle, ultimately leading to muscle contraction. Now the body needs to stop the signal so the muscle doesn't stay contracted forever. This happens mainly through an enzyme called acetylcholinesterase, located in the synaptic cleft. Choline is transported back into the nerve terminal to make new H. With acetylcholine removed, the ion channels close, the muscle repolarizes, and the muscle relaxes. Neuromuscular transmission can be affected by drugs or diseases. Knowing these helps in both exams and clinical practice. Blocks H release, muscle paralysis. Reduce calcium entry, weaken transmission. Inhibit acetylcholinesterase, H buildup, continuous stimulation. Autoimmune destruction of ATH receptors, muscle weakness. Block AH receptors used in anesthesia. Nerve action potential reaches the terminal. H vesicles fuse with the membrane. H is released into the synaptic cleft. H binds to nicotinic receptors on the muscle. Sodium enters end plate potential.